Hello, people.
Hello.
How we all doing?
Doing good.
All right, so before we get started, I just want to let you know I haven't graded the written questions yet on the exam. I'm going to do that right after this class, so it should be done today. Um, any other announcements? I guess that's about it. So as I said in the last class, we're moving off into finally material I really love. So looking forward to intercellular and intracellular signaling. The next chapter, by the way, chapter 9 is really complicated stuff. So be prepared. Gird your loins, as they say. You know what gird your loins means? I saw this in a TikTok, I think. So back in, Roman era or Macedonian era, whoever was trampling over the Middle East at the time, men wore skirts. Everyone wore skirts. And if you went into battle, you didn't want to have to worry about whatever. So it's like you pull your skirt back and then through your legs and tuck it into your belt, and your loins are girded now. Yeah, that's what it means. Pretty crazy. Okay, so it's about time to start. So how about just do a quick review? Someone asked if I could do a quiz before class instead of reviewing the notes. Good idea. I'm just not prepared for that right now. My other classes have been kind of busy last week, so I didn't get to do that. But I think I'm going to do that, give you like one of the more complicated questions, like the written questions on the exam. And then we can sit and work through it so we can talk about how to answer these kinds of questions. So we'll start doing that Wednesday. Today, I just wasn't prepared. I had too many other things to do. Okay, so let's go ahead and fire up the PowerPoints here. Bum, 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 bum. Ooh. Wow, I found my share pretty quick today. I think I may just leave it in this mode rather than the full screen mode. If that's hell, let's go full screen. All right, so in the last lecture, we talked in general about ligands and receptors. Usually your ligand is going to be extracellular because it's hydrophilic. We talked briefly about the hydrophobic ones, but we'll get to that. So ligands, typically they are either small molecules being like amino acids or some metabolite of amino acids. Also proteins, which can be as small as, 3, I think, or 4 amino acids, or as big as, a couple hundred amino acids. And then hydrophobic molecules. And two different types of receptors, your cell surface receptors, which are really the majority of them, and then intracellular receptors for your lipophilic hormones. Types of hormone release. You've got endocrine, where a cell releases it into the bloodstream. The blood then takes it around to everything, and cells with the receptors will respond to it. Paracrine, where you're releasing directly onto the next door neighbor cell. Autocrine, where you're releasing it and also responding to it. And then juxtacrine, where two cells touch each other, and the hormone, more or less, is membrane bound to one cell, and the receptor is in the membrane of the other cell. This is the Notch signaling pathway, which we'll see in chapter 9. Also, the death receptor, I think, is a pretty famous one for this, where you've got cytotoxic T cells that will express Fas ligand, which is like it's a ligand attached to its membrane, so it will touch cells and tell them to die. Wish I had that power. Okay, and as I said, the the hormone goes throughout the body, but only cells with receptors will respond to that. And in addition, the response is going to be based on what receptors are there. Every receptor's got many subtypes. I shouldn't say every, many receptors have several subtypes. Adrenal receptors are a great example of this, but pretty much most of them do. And again, don't memorize this slide, just know that the response in the cell depends on what subtype of receptor is there. And it also depends on what that subtype of receptor is linked to in this particular cell. So it's all very specific to the cell and what receptors the cell is expressing and what those receptors are linked to. Lipophilic hormones, we've got our glucocorticoids like cortisol, and aldosterone is a mineralocorticoid. And then you've got your sex hormones here. They are going to diffuse into the cell and then bind to the receptors intracellularly. We'll see that slide in a sec. Another one is retinoic acid.
Professor Slick,
Yes.
I think we're only seeing slide 1 still.
Oh man.
Oh, there we go. It just switched to slide 10 with retinoic acid.
That's Patrick, right?
Yes.
Patrick, thank you for telling me. When I go to full screen mode, you can't see it, so I'll just leave it in this mode. Thanks for letting me know. Okay, so I don't think I need to back up. It's all kind of review. Retinoic acid. Retinol is vitamin A, basically diffuses into the cell and gets converted into retinoic acid inside the cell. So it's kind of autocrine, although it can also be released and be paracrine. Thyroid hormones, again, lipophilic hormones, they're made by a complicated pathway. The thyroid cell will exocytose thyroglobin into the follicle inside the thyroid. And in there it will be the tyrosines get iodinated, and then it gets endocytosed and broken down into your thyroid hormones. And then different mechanisms of lipophilic hormone receptors. What's important here is the receptor can either be cytosolic, and when the hormone attaches, it goes into the nucleus, or it could be already in the nucleus. The other thing that's important is you've got these chaperone proteins here that are kind of restricting the movement of the receptor until it binds the hormone, and then it can move. What else? Oh, co-activators and co-inhibitors are important here too. So HAT, histone acetyltransferase, is going to help open up that DNA strand so you can transcribe it. Histone deacetylase is going to do the opposite. It's going to cause it to become more condensed. So this becomes a repressor, and this is an activator. So here's your thyroid hormone interacting with both a activator and a repressor. Okay, so then we go into histamine receptors. 4 different types. The most important are H1 and H2, especially clinically. I mean, H1 is going to be involved in the immune response and the mast cell degranulation that we talk about in a second. And H2 are really important for GI problems. If you've got an ulcer or gastric reflux, blocking H2 receptors can relieve that. H3 receptors then are presynaptic. We'll talk about presynaptic receptors today, I believe, if we get that far. And H4 receptors are then involved in this mast cell response. So your mast cells then, the first time your B cell sees an antigen, it makes IgE. The mast cells have got IgE receptors, so IgE sticks to it. The next time you see that antigen, that cross-links and then activates the receptors, which then causes degranulation, which is a release of histamine, and several other things are going to be released. Proteases mostly that are going to break down extracellular matrix. So the histamine released is going to then activate those H4 receptors on different cells and cause chemotaxis of the cell into that area. So the whole idea with this response is you're going to, let's see, I think I have it down here. Okay, so you're activating H1 receptors. This is an H1 response. endothelial cells that make them leaky. So you get more fluid, more cells into the area. And you relax smooth muscle, you get more blood flow into the area. That's why areas of infection look red, because you got more blood flow going in there. And then those H1 receptors are then going to bring in eosinophils and other cells to help with the response. Okay, so that's what we talked about last Wednesday. So let's move on in chapter 8. So the next thing to talk about is a local production of kind of local modulators. I guess you could call them hormones. They're like paracrine signaling molecules. And these are eicosanoids, which are made from, which are made from the breakdown of fatty acids in the membrane. So I talked about this in chapter 2 a little bit when I talked about the phospholipids. So your phospholipid, again, is 2 big fatty acid chains and a polar head group. And at the time I said this second fatty acid here, the one on the middle carbon, the center carbon, is usually arachidonic acid. And that's a 20 carbon fatty acid with 4 unsaturations. So the enzyme phospholipase A2 will break that off, and then it will rapidly be converted into things like prostaglandins, leukotrienes, and epoxides. So these enzymes are there in the cells, and when that arachidonic acid gets released, it rapidly gets turned into this stuff. What else do I want to say? Okay, so here's your pathways. I don't want you to memorize the pathways. What I want you to know from this is that cyclooxygenase will then be important for making prostaglandins and thromboxanes. And these thromboxanes and prostaglandins will be involved in, similar to an H1 receptor, they cause release of or activation of cells that are going to cause kind of inflammation type response, increased pain response, and things like that. So you're increasing blood flow, increasing endothelial cell leakiness, and also increasing pain in the area. And lipooxygenase, leukotrienes, these are used a lot by immune cells of the immune system. Epoxides, I don't really know. I've never heard of these before. Not that important, I guess. So let's not worry about that. Mainly leukotrienes and prostaglandins. By the way, why is it called prostaglandin? It was first found in the prostate gland. Isn't that interesting? Another little factoid I'm throwing out there. Another important thing is this cyclooxygenase. There are 2 forms of it, COX-1 and COX-2. And these are the enzymes that are blocked by your non-steroidal anti-inflammatory agents like aspirin. I think aspirin's classified that way, although nobody uses it anymore. Ibuprofen, acetaminophen, and what's the third one? Oh, God. I forget now. I can see it in my head, but I can't remember the name of it. But anyway, that they block this. And by doing that, they're going to, make your headache go away by affecting blood flow, changing the blood flow to your brain because blood flow is messed up by these local modulators. So you can block that, and it's going to stop the headache, and also blocks pain locally. So that's how your COX-1 and COX-2 inhibitors work. Okay. And here's the pathways. Again, don't memorize this. Maybe someday you'll have to memorize it when you're in med school, but for now, let's just talk about arachidonic being turned into prostaglandins, leukotrienes, and thromboxane down here. Okay, so now the chapter gets into communication between cells in the central nervous system, which I feel a lot more comfortable with. I think this is a really interesting area. And anyway, we've got 2 different, two main kinds of synapses. One is electrical and the other is chemical. So electrical synapses, we've kind of talked about a little bit already when we talked about cardiac cells and the passage of the action potential between cardiac cells. And that's created by these gap junctions. So a gap junction is a connexicon protein. It's a homohexamer, and it forms in the membrane, and then it comes into contact with another connexin from a neighboring cell, and they form a channel between the two. And they are regulated. They can open and close. So those are your gap junctions. And they, again, connect the cells electrically. And when all the cells in a certain tissue are connected electrically, we call them a syncytium. It's kind of an important word. Any word with that many Y's in it has got to be important. So electrical syncytium. And we do have these centrally, I think, but you also have them between cardiac cells, like I say, between endothelial cells and smooth muscle cells. There are all different places where cells are connected by these gap junctions. The other type, which is more common and more important probably for us, are chemical synapses. And in the chemical synapse, basically you've got a presynaptic axon bulb. Have we talked about nerve structure here? I don't think so. But anyway, the axon, at the end of the axon of a neuron, you've got this axon bulb, and that's the business end there. And then on the other side of the synapse, we call this the postsynaptic membrane. This could be the dendrite of a neuron, could be the cell body of a neuron, could be another axon bulb, but or even a muscle cell or something. But basically, it's whatever the post side of the synapse is, the postsynaptic membrane. So we'll talk about what's going on both sides of this membrane today. Let's see, I'm looking at my notes real quick to see if I skipped something. Okay, so in general, this little squiggly blue line represents an action potential. So this electrical signal, the action potential, comes down the synapse or down the axon to the axon bulb, and it's going to cause the release of a chemical, a neurotransmitter, that will diffuse across and then activate receptors on the postsynaptic side. So let's look at that process more closely. So neurotransmitter is going to be stored in these vesicles in the axon bulb. These vesicles are coming from the soma. Everything that gets made in the cell is made in the soma and then transported out to the axons. And these are vesicles with kinesin motors on them that are dragging these vesicles to the end. So we talked about kinesin in chapter 3, attaches to the vesicle and then it walks on along the microtubule. And think about this. I mean, you've got a cell body in the base of your spine, and the axon goes from the base of your spine all the way down to your toes, right? It's a long way to go. And these kinesin motors drag vesicles all the way down there and then put them into the axon bulb. Now, in the axon bulb, these vesicles are going to be attached to microfilaments. So this is microtubules bringing them down here. These are microfilaments that are kind of holding on to the vesicles and keep them in the right position. Remember, nothing just floats around in the cell. Everything's controlled to make it more efficient. So these are attached to cytoskeleton here to keep them in order. Now, here you can see in the legend, you've got an enzyme and an amine pump. So these vesicles have got pumps in them. They're actually transporters. They're hydrogen neurotransmitter transporters that are going to bring neurotransmitter into the vesicle and also precursors for the neurotransmitter. And the enzymes in there are going to convert the precursors into the neurotransmitter. So these vesicles then will get filled up with neurotransmitter. And then they're going to come down and dock against this membrane here, the presynaptic membrane, which is really close to, which is in the synapse and really close to the postsynaptic membrane. Let's take a quick look at that, and I'm going to back up. So here, these are docking proteins here that are holding on to the vesicles, and right next to the vesicles are calcium channels. Voltage-bended calcium channels. And so these Voltschmeter calcium channels are right here next to these Dock vesicles. And when the action potential comes down, that depolarization causes opening of these Voltschmeter calcium channels. Calcium comes in, and then these vesicles will fuse and release their neurotransmitter. And the vesicles behind will move forward. It's almost like soldiers walking in line. The rank of vesicles just comes forward so that the next time an action potential comes, the cell is ready to release more neurotransmitter. So that's the presynaptic side and the release of neurotransmitter. And then, you know, if you're constantly fusing vesicles to this membrane, the membrane is going to get bigger and you're going to run out of vesicles, right? So the cell needs to retrieve this stuff from the membrane. So that's where the endocytosis comes in. You're going to bring that plasma membrane back in along with the pump that's in the membrane there, or the transporter. And then you're going to recycle the vesicle so it can be used again. And we'll talk about neurotransmitter in a second. What's next? Oh, okay. All right, so you've got your vesicles storing neurotransmitter right next to the plasma membrane. And when calcium comes in, they fuse and release. Let's watch that in my stupid little animation here. Okay, can you see this animation, Patrick?
I could see it, but it's not playing.
Okay, I'm going to play it now. So there's calcium coming in, vesicles fusing, releasing neurotransmitter, and the backward rank coming forward.
It's still not playing.
Oh, it's still not playing? All right. Well, it'll be, oops. Let's go back to this. Let's stop sharing and come back. Sorry. Okay, so all right. If you come to this PowerPoint later, you can find this animation. You can pull it up. It's not that big a deal, but it's kind of cool. So basically, when the action potential comes, these Voltschmeter calcium channels open, calcium comes in, and then these vesicles fuse, release their neurotransmitter. They're going to bind to receptors on the postsynaptic membrane, which is going to cause ions to come in and change the membrane potential of the postsynaptic membrane. And at the same time, these later ranks are coming forward to be ready for more release. Okay, so the next thing we need to know about the synapse, the presynaptic membrane, is this guy over here. These are presynaptic autoreceptors. So when the nerve releases neurotransmitter into the synapse, it is going to activate the postsynaptic membrane, but it's also going to activate these presynaptic autoreceptors, which is going to inhibit the calcium channel. And that's going to inhibit further release. So it's kind of a feedback mechanism here in that, you know, if you already have a lot of neurotransmitter out here, there's no point in releasing more neurotransmitter. You've already activated that synapse. So this is blocking it so that doesn't keep happening. You're not wasting neurotransmitter. And this mechanism we'll see in chapter 9. It's basically this receptor is inhibiting the production of cyclic AMP. Cyclic AMP is involved in regulating these calcium channels. And so this isn't going to shut the calcium channel. It's just going to decrease the amount of calcium that comes in for every action potential. And so you're getting less neurotransmitter release. So that's our presynaptic autoreceptor. Now, in terms of fusion of these molecules, this is from your book, fusion of these vesicles. There are a number of proteins that are important here. So as the vesicle comes close to that presynaptic membrane, it's going to block to these tethering proteins, not the snares, these tethering proteins here. Let me see, I think I like my figure better. So you've got a coiled tethering complex and a multi-subunit tethering complex. And they're going to hold on to that vesicle and then prime it and get it right next to the to the membrane. So these are kind of holding on to it, keeping it in place. And then you get the interaction of the snares here. Now snares, you've got two different types of snares. You've got a V snare, V for vesicle, it's on the vesicle. And then you've got T snare, T for target, it's on the target membrane. In this case, we're talking about the plasma membrane. But these tethering complexes are going to bring that down in close. So right now, where we're at here is basically, come on, right, this figure. Okay, we're tethered right next to the plasma membrane, right next to the calcium channels. Okay, so then, let me back up one. When calcium enters this other protein, RAB, let me go forward one, I'm sorry to keep jumping around, but I like this figure better, but I want to show you the one from your book. But anyway, this protein, RAB is another small monomeric G protein, like RAC-RO and CDC42 that we saw in chapter 3, but it's attached to the vesicle here. So when this thing tethers, your RAB GTPase is right there next to the coiled coil, and I mean right next to the snare complexes. So when calcium enters, RAB is going to lock the snares together, and this locked snares causes fusion of the vesicle with the plasma membrane, and it's an energy-dependent process. It takes ATP to do it, but your locked snares here cause that fusion to start, and then it gets released. Now, these proteins are going to be stuck in the membrane here, right? After it fuses, lets go, you still have your locked snares. So we've got these other proteins, NSF and SNAPs, that are going to release those snares from each other. So in this figure, okay, here's RAB. RAB is sealing them together. You get fusion, and then NSF and SNAPs are going to cause them to break apart. And now this can be recycled to the membrane. Recycled to vesicles internally. So, and then reused. So that's our process here of fusion. Basically, your vesicle comes close, it tethers to the plasma membrane. Then when calcium comes in, RAB seals the snares together. That causes fusion of the vesicle. And later NSF and SNAPs are going to release that fusion so you can recycle those and use them again. Okay, and here's a cool picture of what the snares look like when they fuse together. And I believe, let's see, I don't have my notes with me today. I believe there are typically 1 V snare to 3 T snares that are involved in the mechanism. Okay, any questions about that? Sound good? Okay. All right, now, once you get that neurotransmitter released, how does it get taken back into the cell? How do you stop synaptic transmission? So as long as you've got neurotransmitter in the synapse, you're going to have transmission going on. So how do we stop that? Let me see if I can't. Oops. I'm going to bring up this animation. Now I'm going to stop sharing. I'm going to share differently. See if we can see this animation. Oh, come on, man. Come on, baby. That's not it. Cancel. Untitled. My AV's letting me down. Well, here, I think I can share my entire screen. Okay, can you guys see this animation now with the big arrow in the middle of it?
Right now we can see like a notes tab like on your computer.
Yeah, that's not what we need. Boom, bum, 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 boom. Stop sharing. All right, well, I'll just show you the PowerPoint. You can look at the animation later. It's not that big a deal anyway. Okay, so basically the main way neurotransmitters are released, what's most common is you've got an amine pump here that's going to bring that neurotransmitter back in. So as long as you're bringing that neurotransmitter back in, synaptic transmission is going to decrease. This amine pump, getting back to chapter 2 and membrane transport, is a sodium amine cotransporter. So it's a symporter that brings in that. And we thought it was it's called a pump, and typically pumps are only, you only use the word pump when you're talking about an ATPase, but it was years before we realized it was a sodium amino acid transporter, but that's what it is. Similar to what's on this. I don't think it's the same protein, but it's similar in that it's a cotransporter. And this is, again, that pump on the vesicle that's resupplying the vesicle. So in this scenario, basically you release neurotransmitter, gets taken up by the pump, and then it's recycled back into a vesicle. Or some of it can be broken down by the mitochondria. And that, I like to throw in the drugs. This is how your tricyclic antidepressants work, in that they block MAO inhibitors, MAO. They're going to block those, the enzymes that break down neurotransmitters like norepinephrine, dopamine, serotonin. That's going to increase the amount of neurotransmitter, filling your vesicles better, and you can get more neurotransmitter release. That's how those drugs work. As long as we're talking about drugs, let's do some cocaine. So that's what like cocaine and amphetamines do. They block this reuptake inhibitor. And by doing that, you get a lot more neurotransmitter in the synapse. In fact, amphetamines like methamphetamine will cause a reversal of this pump and this pump. So you're basically dumping all your neurotransmitter out into the synapse. Okay, so that's one mechanism of neurotransmitter removal. Another one, which is pretty interesting and complicated, is we see this with glutamate, which is a really important neurotransmitter in the brain. Glutamate released into synapse, will diffuse out of the synapse and then be taken up by transporters into the astrocytes, which are glial cells right there at the synapses that are helping the synaptic transmission happen. So once it gets into the glial cell, it could be broken down there, or it could be transported back into the neuron to be reshuffled into that vesicle and reused again. So that's another way, astrocytes taking the neurotransmitter up. The last slide of this lecture, chapter, I'm going to talk about that a little more, because we know a lot more about glial cells now than we used to, and it's kind of interesting stuff. So that's the second way. And the third way, I believe we've talked about already, is breaking that neurotransmitter down in the synapse. So the only one I can think of that does this is acetylcholine. So you've got acetylcholinesterase, which is a lipid attached to the outside of the membrane of the presynaptic membrane, and it's going to break that acetylcholine down into acetate and choline. Acetate is everywhere in the body, so that's not a big deal, but the choline gets taken back up, and I imagine the acetate too, and then they both get pumped into vesicles, and then you've got your choline acetyltransferase here in the vesicle that's going to make acetylcholine again. By the way, I'm just bringing up choline acetyltransferase because it's one of those enzymes that was named back in the 50s, and the beauty of it is it's named for what it does. It's just so much easier to remember these things when they're named that way. In the next chapter, we're going to get into signal transduction, and things are going to be called smoothened and hedgehog and patched, and it's going to be like, that name tells me nothing. You just have to memorize what it does. But I love an enzyme that tells me what it's going to do. So those are the three different ways that neurotransmitter can be taken up from the synapse. It can be pumped back into the presynaptic membrane. It can diffuse out of the synapse and then it be taken up by astrocytes, or it can be broken down in the synapse. Okay, and I want you to memorize all of this. No, I'm just kidding. I hate memorizing unless it's important, but why did I put this up here? This is from your book. It's just talking about different neurotransmitters. I believe these are mostly in the brain. Glutamate, glycine, GABA, ATP and adenosine, activating purine receptors, purinergic, acetylcholine, norepinephrine, dopamine. I'm just trying to remember why I'm bringing this up. I guess so you can see the different types of ligands. You've got amino acids, nucleotides themselves. That's what caffeine does. It blocks these purinergic receptors and wakes you up. As you get tired, you get more and more release of adenosine in your brain, and that makes you tired, makes you go to sleep. Okay, amines, which are, again, made from amino acids typically, not acetylcholine, but norepinephrine is made from tyrosine, dopamine is made from tyrosine, serotonin is made from tryptophan, histamine from histidine. So these are rapidly made from amino acids. And then we've got our neuropeptides here, which are short or small mostly, but opiates are a little bit bigger. And then our lipids, and the endocannabinoids. I got to talk about them. I haven't yet. I think I'm going to put them into the next chapter because it's not really covered well in your book, and I think it's worth talking about. Okay, so again, don't memorize this. These are just examples of that, similar to, let's go back. Kind of similar to this slide where I'm just talking about them. Here you've got a list of them that you can look at. Okay. All right, so that's it for this chapter. One more thing I want to talk about, which is not covered in your book because it's something, I just read this paper last week and I was like, wow, this blew my mind. So we always think of glial cells as being the helper cells. So neurotransmission is done by the neurons, all the, information is passed by neurons, but then you've got the glial cells that are just maintaining them. Well, like everything in biology, it's more complicated than that. Your glial cells are also passing information. So what happens here, is this is a glutamate neuron, okay? So you're releasing glutamate, activating NMDA receptors. But in addition, here you've got M-glutes and I-glutes, so metabotropic glutamate receptors and inotropic glutamate receptors. These are the NMDAs over here. But you've got these astrocytes there that are taking up the glutamate after it's released, but they're doing more than just taking it up and passing it back over here, like I said earlier. They're also, you also get activation of metabotropic glutamate receptors on these astrocytes. That's going to cause production of IP3, release of calcium, and fusion of vesicles containing neurotransmitters onto that glutamate neuron afterward. So it's so hard to talk about cell biology because it's like I can't teach you one thing because there's 3 other things you need to know that one thing. So in the next chapter, we're going to talk about IP3 and calcium. I believe I've talked about it a little bit previously, but not that much. But it's a classic way that a cell can be stimulated to release some chemicals to cause exocytosis. So just like when calcium came in through the Voltschmidt calcium channels here in this presynaptic neuron and you get fusion, over here, you're activating these metabotropic receptors, which are causing the internal release of calcium and fusion of vesicles here, releasing neurotransmitters here. Okay, so that's the basic mechanism of how it's released. What is being released? D-serine, which is kind of weird because in our cells, we use all L amino acids. So in proteins, they're all made from L amino acids. There is an isomerase in these astrocytes that is going to turn L-serine into D-serine, and that's being used as a gliotransporter here. It's going to bind to receptors in the in the synapse and cause changes in the neurons. We also have release of glutamate from these cells and release of ATP through hemichannels. So hemichannels, these guys here, they're basically half of a gap junction. So this is also kind of new to me and kind of mind-blowing in that here you've got a gap junction protein that's not lined up to another gap junction in a different cell. It's just opening and allowing ATP to be released. So, you know, like I said a moment ago, ATP is another kind of more of a glial transporter, transmitter. It's released and it increases in your brain and it makes you tired, and then you go to sleep and the glial cells will clean all that up. But you know, it's being released through these hemichannels, which is kind of interesting. Okay, any questions about that? That's it for this chapter. I'd like to move on and start the next chapter a little bit, if that's okay by you guys. All good? Patrick, you're my eyes and ears.
That works for me. I can't call it for everybody else, though.
I think you're the line leader. Everyone respects you, and so whatever you have to say, everyone's going to go along with.
Hey, Dr. Slish.
Yes.
I was looking on Brightspace and I didn't see any new lecture slides though.
Let me see. I may not have uploaded them yet. Let me see. Yeah, I haven't uploaded them. All right. Well, do you want to do that now? I can upload it right now.
Yeah, that sounds good. Thank you.
All right, let's do that. Upload files. All right, I'm going to upload the notes first. Now these, I guess I didn't upload them because I'm not done yet. I'm still working on this chapter. But we can get started here, and I'll just update these files as I complete them. Okay. So there are the notes and the PowerPoint. Oops, it's open. Bum, 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 bum. Okay. Okay, I'm going to change my share to chapter 9 now. Bum, 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 bum. Chapter 9. Okay. Cell signaling events. Now this is typical of the kind of figure in your book, which I am not happy with, so I make my own figures. This is a little too much all in one figure to make any sense out of. So we're going to go a little slower and work our way into these signaling pathways a little bit more gently, let's say. So, okay, receptor tyrosine kinases are one broad category of cell surface receptor for hydrophilic hormones. So they're really important in controlling cell division. We'll talk about this in chapter 10, which I believe is where we're going next. And they're also important in controlling differentiation. In terms of early embryo development, holy cow, it's all about receptor tyrosine kinases and also receptor serine kinases and a number of other things, but these are really important in early embryo development. Tissue remodeling, so like you've got hormones that are going to be important for making new blood vessels, angiogenesis, for when there's tissue damage and you need to reestablish a blood supply, things like that. And wound healing, okay. Anytime you've got damage, you need more cells to get in there and replace the bad cells and release collagen to make scars and all this stuff. A lot of this is under control of receptor tyrosine kinases. And also we talked about cell crawling back in chapter 3. And cells crawling at the time we said was controlled by RAC-RO and CDC42, which are small monomeric G proteins. We're going to see how these are activated here today within this mechanism. Now, the hormones that activate these kinases are typically called growth factors. So you've got epidermal growth factor, nerve growth factor, FGF growth factor, vascular endothelial growth factor. They're all kind of blah, blah growth factor. NGF, I don't know why I remember this, but was studying for my prelims back in grad school, and I was memorizing who won all the Nobel Prizes back then. And Rita Montalcini won the Nobel Prize for NGF. So a woman Nobel Prize winner, yay. We like that. All right. Anyway, the receptors. So the receptors for these are structurally similar. They're all structurally similar. You've got an extracellular ligand binding site, a single alpha helix going through the membrane, one transmembrane region, and then you've got an internal domain that's a kinase. And then you've also got on this internal part a whole bunch of tyrosines. And here is the EGF receptor after EGF is bound. And once you bind the hormone to the receptor, two receptors will bind to each other and activate. So that's kind of key to the first part of this mechanism. When the hormone binds to the receptor, they clump together and then they cross phosphorylate. So the kinase domain on one receptor will phosphorylate the tyrosines on the other, and vice versa. So this is really important to the mechanism because this is going to produce what we call SH2 binding domains. So these are domains which are like the phosphorylated tyrosine plus about 4 other amino acids in the area. Like I think they're like upstream from it. So you've got a whole bunch of different SH2 domains that are all linked to phosphotyrosines. And what this is going to do is activate a signal transduction mechanism called recruitment. So the receptor activates itself and then it recruits other enzymes and things to the membrane that then cause the signal transduction to happen. So here you can see this one receptor, the EGF receptor, can bind 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different 10 different proteins. So, you know, it's funny, you know, they talk about what is it? Rocket science being complicated. Cell biology is far more complicated than rocket science. And I say that as a guy who knows nothing about rocket science. But all these are different pathways that can be activated at the same time by this receptor. And we'll only talk about a couple of them. We're going to talk about phospholipase C gamma. We'll talk about the STAT, the JAK-STAT activation. GRAB2 will be important. You know, we'll see SHIC, but it's not that important. PTP. Now this is kind of important. PTP stands for phosphotyrosine phosphatase. So we always have these feedback mechanisms in every receptor pathway. And this is an example of that. When you dimerize these receptors and cross-phosphorylate, you get all these phosphotyrosines. One of the things that's going to be brought to the membrane is an enzyme that's going to take the phosphates off the tyrosines, phosphotyrosine phosphatase. So it's got that built-in negative feedback mechanism. Now, what's interesting about that is when we get into cancer biology, what happens if you don't have PTP? You know, you could have a mutation of the PTP protein, and if it doesn't come on, then you don't shut these receptors off once they get on. So that feedback is really important here. Okay, so but anyway, bottom line, what we want to see, what we want to talk about in this figure is that dimerization of the receptors, cross-phosphorylation, producing these SH2 binding domains. So what the hell is that? So this goes back to a protein I talked about back in chapter 3. This is an integrin, focal adhesion, right? And integrins are these transmembrane proteins in the focal adhesion. And you've got a number of proteins here, mostly structural, talin and vinculin, alpha actin, and things like that. But here are two enzymes, focal adhesion kinase and SRC. SRC was the first oncogene ever found. It was found in Rous sarcoma virus, which is a cancer-causing virus that affects chickens. And so SRC then was found, it does stimulate cell division, basically. And here is what the structure of SRC looks like. It's got 3 domains. S1 domain, SRC homology 1 domain, is a kinase. It's a tyrosine kinase. And then you've got SRC homology 2 domain and SRC homology 3 domain. So SH2 domain is going to bind to these phosphotyrosines that we saw in the last slide. So this SH2 domain is going to have a binding pocket for that phosphorylated tyrosine, but also some amino acids are going to stick out and make sure it's on the right phosphotyrosine. So you've got an SH2 domain that's going to bind to phosphate plus, what is this? Tyrosine, histidine, alanine, and glycine, I think. So it's going to be looking for all those amino acids as well as the phosphorylated tyrosine. So that's what these things do. And there are a lot of different SH2 domains, SRC homology 2. So a lot of domains that have a structural similarity to these, but have got different amino acids in them that are going to bind to different phosphorylated tyrosines. And then our other domain that's going to come in play here is the SH3 domain. And this is another domain that's going to bind to other proteins. It's going to bind to proteins with polyproline regions. So, you know, proline is kind of that weird amino acid that the amino acid backbone is actually part of the side chain of the amino acid. So it causes a little kink in the chain. So if you've got a number of prolines in a row, the amino acids are going to turn in kind of a circle. And so this SH2 domain is looking for that. When you've got a number of prolines in a row, it will bind to that. So these domains then are kind of like cassettes. They're put on different proteins, and they're going to cause recruitment of these proteins to these activated receptors. So let's see how that goes. So this is kind of cartoonish, but it's like it's a cassette. You know, you can plug this into a different protein, and then that protein would bind to that receptor. So your SH2 domain then has got a site for the phosphotyrosine and a site for the flanking amino acids that make it more specific than just any phosphotyrosine. Here's your SH3 domain. You've got a proline-rich region, which is kind of curvy and stuff. So that's going to bind to this SH3 domain. Okay, so why do we care? Well, here are a whole bunch of different proteins that are going to utilize these SH2 and SH3 domains. Here's our friend SRC, okay? So you've got the kinase domain, SH2 and SH3. FPS, I don't know what that is, or psych. I haven't seen these before. There are so many kinases in the cell, but each of them has got their own SH2 domain, right? And that's going to recruit it to a receptor. Here's another negative feedback protein, GAP, guanine, GTPase activating protein. So we'll see as we get into this mechanism that we're going to have G proteins. This is going to cause a G protein to break its GTP down faster and turn it off. So we'll see that shortly, but basically you can see you've got SH2 domains here to bring it up to receptors. Here are SH3 domains that are usually protein-to-protein interactions, keeping the gap attached to something else. Here's our old friend, the plextrin homology domain. Remember that bad boy? That was, oh no, we haven't talked about that yet. I'm sorry. We'll talk about that probably in the next class. But this is going to bind to phosphatidylanosytol trisphosphate. So that's a complicated thing we'll talk about in the next class. Here's phospholipase C gamma. So phospholipase C is a protein that will break down PIP2 and release IP3. Again, we're going to see that in the next couple lectures, but bottom line being you've got these SH2 and SH3 domains that help it to recruit to the membrane. Now here's an interesting one here, GRAB2. And GRAB2 is similar to another one we'll see, SHIC. These are not enzymes. What they are is scaffolding proteins. So what they're going to do is bind to proteins with their SH2 domains and then bind the receptor with the bind to an enzyme with their SH3 domains and bind to the receptor with its SH2 domain. So they're basically adapter proteins that are bringing some other enzyme to the membrane. And we'll see that in the next slide, I believe. Okay, so here's your GRAB2. It's got an SH2 domain that's going to be specific for certain SH2 binding domains in the receptor. It's got two SH3 domains that are going to attach to a protein called SOS. Okay, so SOS. This gets me back to choline acetyltransferase. SOS is a protein whose name will not help you understand what it does. It's SOS stands for son of sevenless, which again, we're gonna talk about this as we get through the rest of this chapter. A lot of these signaling molecules in these receptor tyrosine kinase mechanisms were discovered through Drosophila genetics. Very powerful tool. Basically, they take Drosophila eggs, they zap them with some mutagen, usually UV light, and then they let the organism grow, and then they see which ones are screwed up. And they take those messed up Drosophila and they figure out why are they messed up. So SOS comes from, they did this experiment and they found a certain mutation would cause a difference in the number of ommatidium, they're called eye facets in the eye of the Drosophila. A certain mutation would cause the 7th ommatidium to be lacking, and then another mutation was related to that. And so the one that caused the lack of the 7th ommatidium was called sevenless, and it turned out to be the EGF receptor. And then the other one related to it, they called son of sevenless, because they could tell they were related. So what are we going to call it? Son of sevenless. So basically, son of sevenless was discovered because it was associated with a receptor tyrosine kinase. So that's a long way of saying it's called son of sevenless, SOS. What's important about son of sevenless is less how we found out what it was, but what it does, obviously. So your tyrosine kinase receptor gets autophosphorylates, and that brings GRAB2. GRAB2 brings SOS with it. SOS then is a GEF, guanine nucleotide exchange factor. And this is what's going to activate our G proteins. So G protein here would be like RAC-RO CDC42. In this mechanism, it's going to be RAS. So RAS is a small monomeric G protein, and in the off state, it will have GDP attached to it. When you get a guanine nucleotide exchange factor like SOS come near the membrane, it will kind of stick a little finger inside that and open it up, and the GDP flies out. And then GTP comes in, and now it's activated. So this G protein now in the active form will go bind to other proteins and turn them on. And it will stay like that until it interacts with this guy, GAP, which we just talked about. And GTPase activating protein will then cause this G protein to break its GTP down into GDP and turn it off. So you can imagine a situation where you've turned a receptor on, and that receptor is turning on the G protein, and at the same time, you've recruited this to the membrane, and that turns it off. So while the receptor is activated, you're cycling through on and off states of this G protein, okay? And then when the hormone goes away and the receptor gets turned off, it stops in the GDP form. So again, here's GAP. I just talked about this a minute ago. Back up a tad. GAP. So when you activate these receptors, you also turn on this negative feedback mechanism. which is, oops, sorry, too far. Where the hell am I? Which is going to turn off the G protein. Okay, so when we cross-phosphorylate receptors, we bring up SOS, which is our GEF, to turn on a G protein. And here we can see that here. So here's your receptor, cross-phosphorylates, brings up GRAB2, GRAB2 brings SOS, SOS activates RAS. And once RAS is on, now we're going to start the rest of the mechanism. Backing up just a tad, SHIC I talked about. SHIC is another protein similar to GRAB2 in that it's got an SH2 domain on one side and an SH2 binding domain on the other side. So this can be another adapter between proteins to recruit more enzymes to the membrane. In your book talks about FRS2 alpha. Don't memorize that, but just know that there are these adapter proteins. Okay, so summing up real quick, receptor tyrosine kinase, autophosphorylation, recruiting other enzymes to the membrane, and activating RAS. Now, once RAS gets activated, we're going to activate a kinase cascade. So RAS is going to activate RAF. RAF is I believe, serine kinase. Let me take a look at my notes real quick. RAF is a serine threonine kinase. Let me just pause for a sec to talk about the significance of that. So most kinases in your cells are going to be serine threonine kinases, okay? So they're going to phosphorylate on serines and threonines, which have an OH sticking out. The minority of kinases are going to be like the kinase domains in the receptor, which are going to phosphorylate on tyrosine. So the first kinase we activate is RAF. It is a serine threonine kinase. That will then activate the next one. It's called MEK, and MEK is a serine threonine and tyrosine kinase. It is promiscuous. It will phosphorylate anything you care to give it. So it is going to then activate MAP kinase, and MAP kinase is pretty key here. Once that gets activated, it goes into the nucleus and it will phosphorylate transcription factors, and that's going to cause changes in transcription. So let's back up and look at that again. So we've got this kinase cascade. You activate the G protein, and that activates RAF. RAF activates MEK. MEK activates MAP kinase. Now, when it comes to this is the simplistic way of looking at it, MEK activating MAP kinase. In reality, first, MEK has got several different names it goes by. It could be called MAP kinase, MAPKK. And there are several different isoforms of this in different cells. MAP kinase is kind of a general term, and probably the more specific term would be ERK, ERK, extracellular related kinase, ERK. And there's ERK1 and ERK2. ERK1 and ERK2 are MAP kinase. They're both, like I say, it's kind of a general name for MAP kinase. Okay, but the key is we get activation of MAP kinase. It comes into the nucleus, and we phosphorylate transcription factors. So we don't get into transcription regulation in this class. We could, I guess. I wasn't planning to, but we can. But bottom line is, when you activate a transcription factor, it's going to bind to enhancer regions in the gene, and that's going to turn the gene on. So that's what we're doing here. We're activating gene transcription or blocking gene transcription, depending on what that hormone does. Okay, should I go through this pathway again? Let me just do it real quick from the top. So hormone binds to receptor. Receptors clump together. They transphosphorylate or cross-phosphorylate, and they produce these SH2 binding domains. That attracts proteins with an SH2 domain up to the membrane, and you get this recruitment going on. The recruitment then brings SOS to the membrane. Now it's near RAS. I don't know if you can see this little yellow thing here. RAS has got a lipid attachment, so it stays with the membrane. So we activate RAS by recruiting SOS, RAS then activates RAF, RAF activates MEK, MEP activates MAP kinase, and MAP kinase then completes the signal transduction. Okay. What am I doing here? Feedback. So this is from your book. We've got a lot of feedback regulation of this pathway, of course. Let's see. So here, ERK. So this is MAP kinase, ERK1 and ERK2. Okay, it can be caused different. It can be called either of those. It's going to phosphorylate MEK and inhibit it, phosphorylate RAF and inhibit. So that's negative feedback on the kinase cascade. RSK we don't need to talk about right now. It'll also phosphorylate the receptor itself and turn it off. So we've got these negative feedback pathways here. And then you've also got the situation where when you activate ERK, you turn on the transcription of DUSPs, I guess you would call them, dual specificity phosphatases. So phosphatases are then going to take the phosphates off these proteins to shut them off. So again, whenever you've got a gas pedal in the car, you've got a brake pedal too. So whenever you activate something in the cell, you're simultaneously activating a lot of these negative feedback systems. Here we're talking about directly phosphorylating these kinases and turning them off. You can also indirectly turn on the production of phosphatases that are going to take the phosphates off. We also talked about GAP being activated. That's going to turn off RAS, and PTP, that's going to take the phosphate off the RTKs and shut them off. And again, I'm stressing all of this stuff because when you get into cancer biology, take out any piece of that puzzle and it's going to change the way the cell works and cause it to become, self-sufficient in terms of growth signals. Okay. That's about as far as I wanted to go for today. Any questions? Let me stop sharing. Okay. Can you guys bring your picture up? Turn your cameras on so we can talk to each other. There's David Roy. Looking spiffy as usual. Talak. Patrick, you're my rock, Patrick. You're the only person who talks to me. Okay, that's all I have for today. Do you have any questions? As soon as I'm done here, I'm going to go and start grading your exam, so you should have those results today. Okay, everybody happy? All right, I'm going to call it a day. Have a great afternoon. Study cell biology.
Have a good one.